Hello everyone, welcome to the next lecture in our physiology video lecture series. This particular lecture is titled Endocrinology Part 2. In the previous lecture on endocrinology, we have gone through the basics of hormone synthesis, chemistry, transport, as well as mode of action of various hormones. In this particular lecture, we will look at specific examples of some particular hormones and the glands which secrete them. So let us begin. The first gland we are going to look into is the pituitary gland and the hormones of the pituitary gland. And thus we look into pituitary hormones and their control by the hypothalamus. On the schematic shown right here, we can see the pituitary gland, which is divided into anterior and the posterior halves, with a small section in between known as pars intermedia. So the pituitary gland has two distinct parts, the anterior and the posterior lobes. The pituitary gland, also called the hypophysis, is a small gland that lies in the sella turcica, which is a bone cavity at the base of the brain and is connected to the hypothalamus by the pituitary or hypophyseal stalk. Physiologically, the pituitary gland is divided into two distinct portions. The anterior pituitary, also known as adenohypophysis, and the posterior pituitary, also known as neurohypophysis. The two portions of the pituitary originate from different sources. The anterior pituitary from Rathke's pouch, which is an embryonic invagination of the pharyngeal epithelium, and the posterior pituitary from a neural tissue outgrowth from the hypothalamus. This difference in origin is what explains that the anterior pituitary from the pharyngeal epithelium has epithelioid nature of its cells. That is, anterior pituitary contains cells which are epithelial in nature, and the origin of posterior pituitary from neural tissue explains the presence of large number of glial type cells in this gland. Thus, the majority of the cells present in the anterior and the posterior pituitaries respectively depend on the origin of these tissues during embryonic development. And as anterior pituitary is developed from pharyngeal epithelium, it has epithelioid nature of its cells. And as posterior pituitary comes from neural tissue, it has large glial type cells in its gland. Next, we will look into the important peptide hormones secreted by the anterior pituitary. These include growth hormone, which promotes growth of the entire body by affecting protein formation, cell multiplication, and cell differentiation. Next is the adrenocorticotropin or corticotropin, which controls the secretion of some of the adrenocortical hormones which affect metabolism of glucose, proteins, and fats. There is also the thyroid stimulating hormone, which is known as thyrotropin, which controls the rate of secretion of thyroxine and triiodothyronine by the thyroid gland. And these hormones control the rates of most of the intracellular chemical reactions in our body. We also have prolactin, which promotes mammary gland development and milk production. One of the important things to notice here is that most of the hormones which are secreted by the pituitary gland actually have actually affect other different glands present in the body and thus control the secretion of another hormone, such as in the case of adrenocorticotropin, which control the secretion of adrenocortical hormones. And thus, this is why pituitary gland is also known as the master controller or the master gland that is present in our body, which controls essentially the entire hormonal system. Next, we move on to other important hormones from the anterior and the posterior pituitary. Two separate gonadotropic hormones are also secreted by the anterior pituitary. Gonadotropic means that the hormones affect the gonads, that is the testes in males and ovaries in female. These hormones include the follicle stimulating hormone and the luteinizing hormone. Both the hormones are involved in the controlled growth of ovaries and testes, as well as their hormonal and reproductive activities. Next, we'll look into the two hormones that are secreted by the posterior pituitary. These include the antidiuretic hormone, which is also known as vasopressin, which is something that we have already encountered before during the lecture on renal system. The antidiuretic hormone controls the rate of water excretion into the urine, thus helping to control the concentration of water in the body fluids. Next is oxytocin, which helps express milk from the glands of the breast to the nipples during suckling and possibly helps in the delivery of the baby at the end of gestation. Posterior pituitary hormones are synthesized by cell bodies that are present in the hypothalamus. The bodies of cells that secrete the posterior pituitary hormones are not located in the pituitary gland itself, but are large neurons called magnocellular neurons located in the supraoptic and the paraventricular nuclei of the hypothalamus. The hormones are then transported in the axoplasm of the fibers, nerve fibers, passing from the hypothalamus to the posterior pituitary gland. Next, let us look into the different cell types that are present in the anterior pituitary. Here we have a schematic which shows the various cell types that are present in the anterior pituitary. Using special stains, at least 5 different cell types can be differentiated in the anterior pituitary, which secrete different hormones and have different physiological actions. These 5 cell types are somatotropes, which secrete the human growth hormone, corticotropes, which secrete adrenocorticotropin, thyrotropes, which secrete thyroid stimulating hormone, and gonadotropes, which secrete gonadotropic hormones, which include both the luteinizing hormone LH and follicle stimulating hormone FSH. And finally, the lactotropes, which release prolactin. Next, we look into how the hypothalamus controls pituitary secretion. Almost all secretion by the pituitary is controlled by either hormonal or nervous signals from the hypothalamus. Secretion from the posterior pituitary is controlled by nerve signals that originate in the hypothalamus and terminate in the posterior pituitary. In contrast, secretion by the anterior pituitary is controlled by hormones called hypothalamic releasing and hypothalamic inhibitory hormones, which are secreted within the hypothalamus itself and then conducted, as shown in this figure, to the anterior pituitary through minute blood vessels called the hypothalamic hypophyseal portal vessels. In the anterior pituitary, these releasing and inhibitory hormones act on glandular cells to control their secretion. Next, we'll look into the hypothalamic releasing and inhibitory hormones that control anterior pituitary secretion. The function of releasing and inhibitory hormones is to control secretion of the anterior pituitary hormones. For most of the anterior pituitary hormones, it is the releasing hormones that are important, but for prolactin, a hypothalamic inhibitory hormone probably exerts more control. The major hypothalamic releasing and inhibitory hormones are as follows. The first is thyrotropin releasing hormone, which causes release of thyroid stimulating hormone. Corticotropin releasing hormone, which causes release of adrenocorticotropin. Growth hormone releasing hormone, which causes release of growth hormone, and growth hormone inhibitory hormone, which is also called somatostatin, which inhibits release of growth hormone. Gonadotropin releasing hormone, which causes release of two gonadotropic hormones, luteinizing hormone and follicle stimulating hormone, and prolactin inhibitory hormone, which causes inhibition of prolactin secretion. In this particular table, we have summarized the various hypothalamic releasing and inhibitory hormones and their structures. So we can see that thyrotropin releasing hormone is only a peptide of 3 amino acids, whereas prolactin inhibitory hormone and growth hormone releasing hormone are single chain protein peptides of 44 amino acids or larger. Thus, the hypothalamic releasing and inhibitory hormones are varied in nature, but they all have the function of either stimulating or inhibiting secretion of various other hormones by the anterior pituitary. Next, we look into the physiologic functions of growth hormone. All the major anterior pituitary hormones, except for growth hormone, exert their principal effects by stimulating target glands, including thyroid gland, adrenal gland, ovaries, testes, and mammary glands. Growth hormone, in contrast, does not function through a target gland but exerts its effects directly on all or almost all tissues of the body. So let us look into how growth hormone promotes growth of many body tissues. Growth hormone, also called somatotropic hormone or somatotropin, is a small molecule that contains 191 amino acids in a single chain and has a molecular weight of 22,005 daltons. It causes growth of almost all tissues of the body that are capable of growing. It promotes increased sizes of cells and increased mitosis with development of greater number of cells and specific differentiation of certain types of cells such as bone growth cells and early muscle cells. The, the plot that is given here shows typical weight charts of two growing littermate rats, one of which received daily injections of growth hormone and the other which did not receive any growth hormone. This figure shows enhancement of growth in the rat given the growth hormone in the early days of life and even after the two rats reached adulthood. In the early stages of development, all organs of the treated rat increased proportionately in size. After adulthood was reached, many of the bones stopped lengthening, but many of the soft tissues continued to grow, which is why we see a continued rise in body weight. Next, we look into growth hormone and its various physiologic characteristics. Growth hormone has several metabolic activities. Aside from its general effect in causing growth, growth hormone has multiple specific metabolic effects such as increased rate of protein synthesis in most cells of the body, increased mobilization of fatty acids from adipose tissue, increased free fatty acids in the blood, and increased use of fatty acids for energy, and decreased rate of glucose utilization throughout the body. Thus, in effect, growth hormone enhances body protein, uses of fat stores, and conserves carbohydrate. Next, we look into how exactly is growth hormone secretion regulated. For many years, it was believed that growth hormone was secreted primarily during the period of growth, but then disappeared from the blood at adolescence. This has been proven wrong. After adolescence, secretion decreases slowly with aging, finally falling to about 25% of the adolescent level in very old age. Several factors related to a person's state of nutrition or stress are known to stimulate the secretion. These include starvation, which is accompanied by severe protein deficiency, hypoglycemia or low concentration of fatty acids in the blood, exercise, excitement, and trauma. Next, let us look into growth hormone and somatomedins. Growth hormone exerts much of its effect through intermediate substances called somatomedins, also called insulin-like growth factors. It has been found that growth hormone causes the liver, and to a much less extent other tissues, to form several small proteins called somatomedins that have a potent effect of increasing all aspects of bone growth. Many of the somatomedin effects on the growth are similar to the effects of insulin on growth. Therefore, the somatomedins are also called insulin-like growth factors. At least four somatomedins have been isolated, but by far the most important of these is somatomedin C, which is also called IGF-1. The pygmies of Africa have a congenital inability to synthesize significant amounts of somatomedin C. Therefore, even though their plasma concentration of growth hormone is either normal or high, they have diminished amounts of somatomedin C in the plasma, and this apparently is the reason for the small stature of these people. Next, let us look into abnormalities of growth hormone secretion. These abnormalities include first, panhypopituitarism. This panhypopituitarism means decreased secretion of all of the anterior pituitary hormones. The decrease in secretion may be congenital, that is present at birth, or it may occur suddenly or slowly at any time during life. most often resulting from a pituitary tumor that destroys the pituitary gland. Next is dwarfism. Most instances of dwarfism result from generalized deficiency of anterior pituitary secretion, that is hypopituitarism during childhood. In general, all parts of all physical parts of the body develop in appropriate proportion to one another, but the rate of development is greatly decreased. Next is gigantism. Occasionally, the acidophilic growth hormone producing cells of the anterior pituitary gland become excessively active, and sometimes even acidophilic tumors occur in the gland. As a result, large quantities of growth hormone are produced. All body tissues grow rapidly, including the bones. If the condition occurs before adolescence, before epiphyses of the long bones have become fused with the shafts, height increases so that the person becomes a giant up to 8 feet tall. So during growing up, there is a particular stage during which the epiphysis of our long bones becomes fused with the shaft of our bones. That is, this is the point at which we essentially stop growing in height. If gigantism occurs before this point is reached, then the bones grow even larger before fusing. This in turn causes the person to be extremely tall, close to 7 to 8 feet tall, and thus changes them to a giant in case of this abnormality. Next, we look into acromegaly. If an acidophilic tumor occurs after adolescence, that is, after the epiphyses of the long bones have fused with the shafts, the person cannot grow taller, but the bones can become thicker and the soft tissues can continue to grow. This condition is known as acromegaly. Enlargement is especially marked in the bones of the hands and the feet and in membranous bones, including the cranium, nose, bosses on the forehead, supraorbital ridges, lower jawbone, portions of the vertebra, because their growth does not cease at adolescence, and thus the increased release of growth hormone can affect these bones. Next, we look into the posterior pituitary gland and its relation to the hypothalamus. The posterior pituitary gland, also called the neurohypophysis, is composed mainly of glial-like cells called pituicytes. The pituicytes do not secrete hormones. They act simply as supporting structure for large numbers of terminal nerve fibers and terminal nerve endings from the nerve tracts that originate in the supraoptic and paraventricular nuclei of the hypothalamus as shown in the figure. So this is the paraventricular nucleus right here and supraoptic nucleus, followed by which affect the posterior pituitary that is shown right here. And here is the hypothalamic hypophyseal tract, which helps in the transport of the hormones released in the hypothalamus to the posterior pituitary. These tracts pass to the neurohypophysis through the pituitary stalk. It is also known as the hypophyseal stalk. The nerve endings are bulbous knobs that contain many secretory granules. These endings lie on the surfaces of capillaries where they secrete two posterior pituitary hormones, which are the antidiuretic hormone ADH, also called vasopressin, and oxytocin. Next, we look into the physiologic effect of ADH and the oxytocin hormone. First, we look into the physiological functions of ADH. ADH causes decreased excretion of water by the kidneys, as we have already seen in our previous lecture on renal system. In the presence of ADH, the permeability of the collecting ducts and the tubules to water increases greatly, and this allows most of the water to be reabsorbed as tubular fluid passes through these ducts, thereby conserving water in the body and producing very concentrated urine. It functions by mobilizing more aquaporins or water channels to the membrane of the cells lining the collecting ducts, and thus the greater amount of aquaporins mean the increased transport of water, and this in turn is what causes the conservation of water as more water is picked up by the cells. Next, we look at to the functions of oxytocin hormone. Oxytocin causes contraction of the pregnant uterus. The hormone oxytocin, in accordance to its name, powerfully stimulates contraction of the pregnant uterus, especially towards the end of gestation. Therefore, many obstetricians believe that this hormone is at least partially responsible for causing birth of the baby. Oxytocin also aids in milk ejection by the breasts. Oxytocin also plays an essentially important role in lactation. In lactation, oxytocin causes milks to be expressed from the alveoli into the ducts of the breasts so that the baby can obtain it by suckling. Next, we move on from the pituitary gland and its hormones to other glands in the body, starting with the thyroid gland and thyroid metabolic hormones. The thyroid secretes two major hormones, thyroxine and triiodothyronine, commonly called T4 and T3 respectively. Both of these hormones profoundly increase the metabolic rate of the body. Thyroid secretion is controlled primarily by thyroid stimulating hormone TSH, which is secreted by the anterior pituitary gland, as we have already seen before. In the first diagram right here, we see a cross section of the thyroid gland in which we can see the follicle cells, cuboidal epithelial cells, the red blood cells, and the colloid that is present in the thyroid gland. This will come into play and we'll understand its role as we move along. First, we look into the synthesis of thyroid metabolic hormones. Iodine is required for formation of thyroxine. To form normal quantities of thyroxine, about 50 milligrams of ingested iodine in the form of iodides are required each year, or about 1 milligram per week. To prevent iodine deficiency, common salt is iodized with about 1 part sodium iodide to every 100,000 parts of sodium chloride, so that our iodine requirement for formation of thyroxine is fulfilled. Next important step in synthesis of thyroid metabolic hormones is the iodide pump, which is involved in iodide trapping. The basal membrane of the thyroid cell has the specific ability to pump the iodide actively to the interior of the cell. This is called iodide trap. Part of the iodide trapping process is shown here, where in this second diagram, where iodide in the form of I minus is transported into the cells by various proteins, which is then utilized by the Golgi and the ER in order to add the iodine moiety to the tyrosine residue in order to form the thyroid hormones thyroxine and triiodothyronine. Next, we look into thyroglobulin and chemistry of formation of thyroxine and triiodothyronine. We first look into the formation and secretion of thyroglobulin by thyroid cells. In the thyroid cells, a large glycoprotein is formed and secreted, which is called thyroglobulin. Each molecule of thyroglobulin contains about 70 tyrosine amino acids, and they are the major substrates that combine with iodine to form the thyroid hormones. Thus, the thyroid hormones form within the thyroglobulin molecule. Thus, the thyroglobulin molecule forms a framework on which new thyroxine molecules are added. Next, we look into iodination of tyrosine and formation of the thyroid hormones, which is also known as organification of thyroglobulin. The binding of iodine with the thyroglobulin molecule is called organification of thyroglobulin. In the thyroid cells, the oxidized iodine is associated with an iodinase enzyme that causes the process to occur within seconds or minutes. Tyrosine is first iodized to monoiodotyrosine and then to diiodotyrosine. Then, during the next few minutes, hours, or even days, more and more of iodotyrosine residues become coupled with each other. The major hormonal product of this coupling reaction is the molecule thyroxine that remains part of the thyroglobulin molecule. So here we have a schematic showing us the reactions that follow. Tyrosine is first iodine is added to tyrosine by the iodinase enzyme to form monoiodotyrosine and diiodotyrosine. These are conjugated and coupled together to form other iodotyrosine macromolecules and also thyroxine, which still remains attached to the thyroglobulin molecule as iodine is attached to it. Next, we look into storage of thyroglobulin. After synthesis of the thyroid hormone has run its course, each thyroglobulin molecule contains up to 30 thyroxine molecules and a few triiodothyronine molecules. In this form, the thyroid hormones are stored in the follicles in an amount sufficient to supply the body with its normal requirements of thyroid hormones for 2 to 3 months. Next is the release of thyroxine or T4 and triiodothyronine or T3 from the thyroid gland. Thyroglobulin itself is not released into the circulating blood in any measurable amounts. Instead, thyroxine and triiodothyronine must first be cleaved from the thyroglobulin molecule. Then these free hormones are released. The process occurs as follows. The apical surface of the thyroid cells sends out pseudopod extensions that close around small portions of the colloid to form pinocytic vesicles that enter the apex of the thyroid cell. Then lysosomes present in the cell cytoplasm immediately fuse with these vesicles to form digestive vesicles containing digestive enzymes from the lysosomes mixed with the colloid. Multiple proteases among the enzymes digest the thyroglobulin molecules and release thyroxine and triiodothyronine in free form. These then diffuse through the base of the thyroid cells into the surrounding capillaries. Thus, the thyroid hormones are released into the blood. The whole process is shown in the schematic right here. Next, we move on to the physiologic functions of the thyroid hormone. The most important function of the thyroid hormones is to increase cellular metabolic activity. This is done by first increasing the number of activity of mitochondria. When thyroxine or triiodothyronine is given to an animal, the mitochondria in the most of the cells of the animal's body increase in size as well as number. Furthermore, the total membrane surface area of the mitochondria increases almost directly in proportion to the increased metabolic rate of the whole animal. Thyroid hormones also increase active transport of ions through cell membranes. One of the enzymes that increases its activity in response to thyroid hormones is the sodium potassium ATPase pump. This in turn increases the rate of transport of both sodium and potassium ions through the cell membrane of some tissues. Because this process uses energy and increases the amount of heat produced in the body, it has been suggested that this might be one of the mechanisms by which thyroid hormone increases the body's metabolic rate. Next, we look into the effects of thyroid hormone on specific bodily mechanisms. First, we look into the stimulation of carbohydrate metabolism by the thyroid hormones. Thyroid hormone stimulates almost all aspects of carbohydrate metabolism, including glucose uptake, enhanced glycolysis, enhanced gluconeogenesis, increased rate of absorption from the GI tract, and increased insulin secretion. Next is stimulation of fat metabolism. Essentially, all aspects of fat metabolism are also enhanced under the influence of thyroid hormone. In particular, lipids are mobilized rapidly from the fat tissue. This increases the free fatty acid concentration in the plasma and greatly increases the oxidation of free fatty acids by the cells. Next, look into increased requirements for vitamins. Thyroid hormone increases the quantity of many bodily enzymes because vitamins are essential parts of some enzymes or coenzymes. Thyroid hormone also causes increased need for vitamins. There is also an increased basal metabolic rate in response to thyroid hormone secretion. Because thyroid hormone increases metabolism in almost all cells of the body, excessive quantities of the hormone can occasionally increase the basal metabolic rate 60 to 100% above the normal one. Next is decreased body weight. Greatly increased thyroid hormone almost always decreases the body weight, and greatly decreased hormone almost always causes increase in body weight. Effect of thyroid hormone on the cardiovascular system. Increased blood flow and cardiac output, increased heart rate, and increased heart strength are effects of thyroid hormone on the cardiovascular system. The mean arterial pressure usually remains about normal after administration of thyroid hormone. Because of increased blood flow through the tissues between heartbeat, the pulse pressure is often increased, with the systolic pressure elevated in hyperthyroidism 10 to 15 millimeters of mercury, and diastolic pressure increased to corresponding amount. Next is the increased respiration. The increased rate of metabolism increases the utilization of oxygen and formation of carbon dioxide. These effects activate all the mechanisms that increase the rate and depth of inspiration. Increased gastrointestinal motility. In addition to increased appetite and food intake, thyroid hormone increases both the rate of secretion of digestive juices and the motility of gastrointestinal tract. Excitatory effects on the nervous system. The hypothyroid individual is likely to have extreme nervousness and many psychoneurotic tendencies, such as anxiety complexes, extreme worry, and paranoia. Effect on function of muscles. Slight increase in thyroid hormone usually makes the muscles react with vigor. But when quantity of hormone becomes excessive, the muscles become weakened because of extreme protein catabolism. These were the effects of the thyroid hormone on the metabolic system. Next, we look into the feedback loop which causes the thyroid hormone to decrease anterior pituitary secretion of thyroid stimulating hormone. So what happens is that thyroid stimulating hormone, which is released on the anterior pituitary, acts on the thyroid, which promotes secretion of thyroxine. Thyroxine, when the increased amount of thyroxine increases metabolism of cells, which in turn they affect the hypothalamus. The effect on hypothalamus causes an increased release in thyrotropin releasing hormone, which inhibits the release of TSH by the anterior pituitary. And thus, the whole system essentially is a feedback loop involving the three hormones: thyroid stimulating hormone, thyroxine, and thyrotropin releasing hormone, which act in tandem so that there is controlled release of thyroxine by the thyroid gland. Next, we look into diseases of the thyroid. First is hyperthyroidism. In most patients with hyperthyroidism, the thyroid gland is increased to 2 to 3 times normal size, with tremendous hyperplasia and infolding of the follicular cell lining into the follicles, so that the number of cells is also greatly increased. Also, each cell increases its rate of secretion several folds. The symptoms of hyperthyroidism are a high state of excitability, intolerance to heat, increased sweating, mild to extreme weight loss, which can increase to sometimes as much as 100 pounds, varying degrees of diarrhea, muscle weakness, nervousness or other psychic disorders, extreme fatigue but inability to sleep, and tremors of the hand. Next, we look into causes of hyperthyroidism, which is also known as toxic goiter, thyrotoxicosis, and Graves' disease. In all patients, substances that have similar action to TSH is present in the blood. These substances are called thyroid stimulating immunoglobulins, or TSI, which bind to the same receptor as TSH and stimulate the thyroid gland for as long as 12 hours as compared to just 1 hour by TSH. The antibodies that cause hyperthyroidism almost certainly occur as a result of autoimmunity that has developed against the thyroid tissue. Presumably, at some time in history of the person, an excess of thyroid cell antigen was released from the thyroid cells, and this has resulted in the formation of antibodies against the thyroid gland itself. And thus, hyperthyroidism is essentially an autoimmune disease which is caused by the thyroid stimulating immunoglobulins. Next, we look into hypothyroidism. The effect of hypothyroidism in general are opposite to those of hypothyroidism. But there are few physiologic mechanisms peculiar to hypothyroidism. Hypothyroidism, like hyperthyroidism, probably is initiated by autoimmunity against the thyroid gland, but immunity that destroys the gland rather than stimulating it. Thyroid glands of most of these patients first have autoimmune thyroiditis, which means inflammation of the thyroid. This causes progressive deterioration and finally fibrosis of the gland with resultant diminished or absent secretion of thyroid. Several other types of hypothyroidism also occur, often associated with development of enlarged thyroid glands called thyroid goiter. Next, we look into the physiologic characteristics of hypothyroidism. Hypothyroidism can occur both in patients with iodine deficiency, endemic colloid goiter, which is also known as endemic colloid goiter, and without iodine deficiency, which is known as idiopathic non-toxic colloid goiter as well. The physiologic effects of hypothyroidism include fatigue and extreme somnolence with sleeping up to 12 to 14 hours a day, extreme muscular sluggishness, slowed heart rate, decreased cardiac output, decreased blood volume, and sometimes increased body weight, constipation, mental sluggishness, and failure of many trophic functions in the body, evidenced by depressed growth of hair and scaliness of the skin, development of frog-like husky voice, and in severe cases, development of edematous appearance throughout the body called myxedema. Edematous appearance means the storage or of extracellular fluid, excess amount of extracellular fluid in the interstitial spaces, which causes inflammation of these interstitial spaces, which can be seen from outside. This phenomenon is known as myxedema. Next, we move on to the adrenocortical hormones. The two adrenal glands, which weigh about 4 grams each, lie at the superior poles of the two kidneys. Each gland is composed of two distinct parts the adrenal medulla and the adrenal cortex. The adrenal medulla, the central 20% of the gland, is functionally related to the sympathetic nervous system. It secretes the hormones epinephrine and norepinephrine in response to sympathetic stimulation. The adrenal cortex secretes an entirely different group of hormones called corticosteroids. These hormones are all synthesized from the steroid cholesterol and all have similar chemical formulas. Let us now look into the corticosteroids, which include the mineralocortical steroids, glucocorticosteroids, and androgens. The two major types of adrenocortical hormones, the mineralocorticoids and glucocorticoids, are secreted by the adrenal cortex. In addition to these, small amounts of sex hormones are secreted, especially androgenic hormones, which exhibit about the same effects in the body as the male sex hormone testosterone. In this particular schematic, we have shown a cross-sectional view of the adrenal glands, which include the zona glomerulosa, which secrete aldosterone, The zona fasciculata and the zona reticularis, which release cortisol and androgens, and the medulla, which release catecholamines. Next, we look into the synthesis and the secretion of adrenocortical hormones. The adrenal cortex has three distinct layers. The zona glomerulosa, a thin layer of cells that lies just underneath the capsule and constitutes about 15% of the adrenal cortex. These cells are the only ones in the adrenal gland capable of secreting significant amounts of aldosterone because they contain the enzyme aldosterone synthase. The zona fasciculata, which is the middle and the widest layer, constitutes about 75% of the adrenal cortex and secretes the glucocorticoids, cortisol, and corticosterol, as well as small amounts of adrenal androgens and estrogens. Next is the zona reticularis, which is a deep layer of the cortex and secretes the adrenal androgens, DHEA, and androstenedione, as well as small amounts of estrogens and glucocorticoids. Next, we look into the synthetic pathways for adrenal corticoids, adrenal steroids. The figure gives the principal steps in the formation of important steroid products of the adrenal cortex, aldosterone, cortisol, and the androgens. Essentially, all these steps occur in two of the organelles of the cell, the mitochondria and the endoplasmic reticulum. Some steps occurring in one of these organelles and some in the other. Each step is catalyzed by a specific enzyme system. So the starting point for all of the adrenocortical hormones is cholesterol, which after various enzymatic reactions gives rise to aldosterone, cortisol, and the androgens. Next, we look into the functions of the mineralocorticoids. They are looking especially at aldosterone. First, we look into the renal and the circulatory effects of aldosterone. Aldosterone increases renal tubular reabsorption of sodium and secretion of potassium. Aldosterone increases absorption of sodium and simultaneously increases secretion of potassium by the renal tubular epithelial cells, especially in the principal cells of the collecting tubules, and to a lesser extent in the distal tubules and the collecting ducts. Therefore, aldosterone causes sodium to be conserved in the extracellular fluid while increasing potassium excretion in the urine. Next, we look into the cellular mechanism of aldosterone action. First, because of its lipid solubility in the cellular membranes, aldosterone diffuses readily to the interior of the tubular epithelial cells. Second, in the cytoplasm of the tubular cells, aldosterone combines with a highly specific cytoplasmic receptor protein. Third, the aldosterone receptor complex diffuses into the nucleus where it induces formation of messenger RNA related to the process of sodium and potassium transport. This whole mechanism of action is similar between all steroid hormones and is something that we have discussed in the previous lecture. Next, we look into function of glucocorticoids. At least 95% of glucocorticoid activity of the adrenocortical secretions results from the secretion of cortisol, also known as hydrocortisol. In addition to this, a small but significant amount of glucocorticoid activity is provided by corticosterol. First, we look into the effects of cortisol on carbohydrate metabolism. Looking into stimulation of gluconeogenesis. By far, the best metabolic effect of cortisol and other glucocorticoids on metabolism is their ability to stimulate gluconeogenesis, which is the formation of carbohydrate and proteins and some other substances by the liver. Cortisol increases the enzymes required to convert amino acids into glucose in the liver. This results from the formation of glucocorticoids to activate DNA transcription in the liver cell nuclei in the same way aldosterone functions in renal tubular cells, with formation of messenger RNAs that in turn lead to an array of enzymes required for gluconeogenesis. Cortisol causes mobilization of amino acids from the extrahepatic tissues, mainly from muscles. As a result, more amino acids become available in the plasma to enter into the gluconeogenesis process of the liver and thereby to promote the formation of glucose. Decreased glucose utilization by the cells. Cortisol also causes moderate decrease in the rate of glucose utilization by most of the cells of the body. Next, we look into effects of cortisol on the protein metabolism. Reduction in cellular protein. One of the principal effects of cortisol on the metabolic systems of the body is reduction of protein stores in essentially all body cells except those of the liver. This is caused by both decreased protein synthesis and increased catabolism of protein already in cells. Next is the increased blood amino acids, diminished transport of amino acids into extrahepatic cells, and enhanced transport into hepatic cells. Studies in isolated tissues have demonstrated that cortisol decreases amino acid transport into muscle cells and perhaps also into other extrahepatic cells. Next, we look into the effect of cortisol on fat metabolism. Mobilization of fatty acids. The effect of cortisol promotes mobilization of fatty acids in adipose tissue. This increases the concentration of free fatty acids in the plasma, which also increases their utilization for energy. Cortisol also seems to have direct effect to enhance the oxidation of fatty acids in the cells. The increased mobilization of fats by cortisol combined with increased oxidation of fatty acids in the cells helps shift the metabolic systems of the cells from utilization of glucose for energy to utilization of fatty acids in times of starvation or other stresses. Obesity caused by excess cortisol. Despite the fact that cortisol can cause a moderate degree of fatty acid mobilization from adipose tissues, many people with excess cortical secretion develop a peculiar type of obesity with excess deposition of fat in the chest and head regions, giving rise to a buffalo-like torso and a rounded moon face. Although the cause is unknown, it has been suggested that this obesity results from excess stimulation of food intake, with fat being generated in some tissues of the body more rapidly than it is mobilized and oxidized. Next, we look into regulation of cortisol secretion by adrenocorticotropic hormones from the pituitary gland. Adrenocorticotropic hormone or ACTH stimulates cortisol secretion. Unlike aldosterone secretion by zona glomerulosa, which is controlled mainly by potassium and angiotensin acting directly on the adrenocortical cells, almost no stimulation have direct control effects on the adrenal cells that secrete cortisol. Instead, in secretion of cortisol is controlled almost directly by ACTH secreted by the anterior pituitary gland. This hormone, also called corticotropin or adrenocorticotropin, also enhances the production of adrenal androgens. ACTH secretion is controlled by corticotropin releasing factor from the hypothalamus. In the same way that other pituitary hormones are controlled by releasing factors from the hypothalamus, an important releasing factor also controls ACTH secretion. This is called corticotropin releasing factor. It is secreted into the primary capillary plexus of the hypophyseal portal system in the median eminence of the hypothalamus and then carried to the anterior pituitary gland where it induces ACTH secretion. Next, we look into the inhibitory effect of cortisol on hypothalamus and on the anterior pituitary to decrease ACTH secretion. Cortisol has direct negative feedback effects on the hypothalamus to decrease the formation of CRF and the anterior pituitary gland to decrease the formation of ACTH. Both of these feedbacks help regulate the plasma concentration of cortisol. That is, whenever the cortisol concentration becomes too great, the feedbacks automatically reduce the ACTH towards a normal control level. Finally, we look into a summary of the cortisol control system. The figure here shows an overall system for control of cortisol secretion. The key to this control is the excitation of hypothalamus by different types of stress. Stress stimuli activate the entire system to cause rapid release of cortisol, and the cortisol in turn initiates a series of metabolic effects directed towards releasing the damaging nature of stressful state. There is also direct feedback of the cortisol to both the hypothalamus and the anterior pituitary gland to decrease concentration of cortisol in the plasma at times when body is not experiencing stress. However, the stress stimuli are prepotent ones. That is, they can always break through this direct inhibitory feedback of cortisol, causing either periodic exacerbations of cortisol secretion at multiple times during the day or prolonged cortical secretion in times of chronic stress. This was all we had for today. These are the references to the slides, which include the chapters 75, 76, and 77 from the textbook of Medical Physiology by Arthur C. Guyton, the 11th edition. I thank you for listening to this video.